Welcome to Grow with Biology. Today we'll deal with apoptosis. Now, what is apoptosis? It is programmed cell death. Means it is a programmed event which results to cell death. The term was coined by John Kern, Adroville, and A.R. Corey in 1972. It has a different steps, and all the steps compiled to the death of a single cell. It is a neat and orderly process. Now it is compared with necrosis, which is not an orderly process, whereas apoptosis is an ordered process or a programmed one. Now, why and how a cell dies? A cell dies due to some of the reasons. First reason is if the DNA is fragmented. We all know the DNA is the genetic material of a cell and all the genes are present inside the DNA. Now, if that DNA has been fragmented, chromatin has been condensed, and this will result in fragmentation of nucleus. Means the DNA nucleus will start forming small circular blips, which will lead to fragmentation of the cell. Now, due to the damage in the DNA, that is by fragmentation, it leads to the damage of the entire cell. As there is no nucleus, so the cell can't survive and the cell has to die. Now, in this mean event, when we are talking about apoptosis, one of the main term which we have to remember is phosphatidylserine. Now, phosphatidylserine is present in the inner layer of the plasma membrane of all the cells. It is compulsorily present in the inner layer. But what happens when the cell is about to die? When the cell has an apoptotic signal, then this phosphatidylserine moves towards the outer layer of the phosphatidyl layer. Now, phospholipid has two layers. The inner layer has phosphatidylserine, but as the cells moves for apoptosis, it flip out or the outer layer. Now, when we talk about C. elegans, there are two genes which we talk specifically is, first is CED4 and second is CED3. Now, these two genes have the property of apoptosis, means CED4 activates CED3, which leads to apoptosis, that is a programmed death. And CED9 is anti-apoptotic, means in normal cases when a cell has to die, then CED4 must activate CED3, resulting in a normal program death. But in presence of CED9, CED9 will inhibit the function of CED4. When 9 is suppressing the activity of CED4, then CED3 cannot be activated. So, if CED9 is active, then there is no cell death. No cell death. Here with cell death is apoptosis. So, CED9 is anti-apoptotic. In mammalian homologs, when we compare, CED4 is known as APOP1 and CED9 is BCL2 family. So, BCL2 is anti-apoptotic in mammalian cells and apoptotic is CED4. Caspases, another term which is related with apoptosis. Now, they are proteases. Proteases means they will cleave the protein. Now, the caspases have cysteine at its own active site, and it cleaves aspartic acids. It will cleave after every aspartic acid in the substrate. So whenever it has to cleave, it will search aspartic acid and then cleave, and it must have its own cysteine in its active site. Now, caspases, when they are synthesized inside the cells, they are present in inactive form. They are not active, so they have to be activated. How they they become active? They activate when there is proteolytic cleavage. Some part or some portion of this protein, when removed from it, then it becomes activate. Now when it has activated, it will inhibit the DNAs. Now when it is inhibiting the DNAs, then they will get activated. Now what is caspase 2? Caspase cleave the nuclear lamins. Nuclear lamins are present in the as a support for the plasma membrane. Nuclear support for the nucleus, as when it has cleaved the backbone, then it will result in fragmentation of nucleus, which will result in disruption of cytoskeleton. There is membrane blibbing and resulting in cell fragmentation, which results in cell death. Now, type of caspases, there are initial two types. One is initiator, another is effector. There are many examples. Some have dealt only with one of the example. The initiator is caspase 9. The effector is caspase 3. Initiator means first uh, caspase which will start the process of apoptosis is the initiator. Once it has initiated, then rest of the caspase will start its activity. So those are known as effector. Now first a signal will attack the cell and it will activate the initiator caspase, which will activate the activator caspase or the effector caspase, and then the cellular target proteins are targeted. When the cellular proteins are targeted, then it starts degrading it. Now, what are the targets of caspases? Now, first is protein kinases. Now, example is FAK, focal adhesion kinase. Now, focal adhesion kinase inactivate and disrupt the cell adhesion. Focal adhesion kinase will help in adhesion, and now when this caspase act on it, it starts making it inactivated. Fact, inactivated means there is no adhesion between the cells, so it disrupts the cell adhesion. Second property is lamins. It has cleaved the lamin, the disassembly of the nuclear lamina, and the nucleus will shrink and then finally broke off. Proteins of the cytoskeleton. Cytoskeletons are cleaved, so the cell will again change the shape. Caspases activated DNA, thus resulting in DNA degradation. These are the four steps where caspase hits, and all these results in final cell death. Now the pathways, there are two pathways. One is intrinsic, another is extrinsic. Intrinsic is due to the internal stimulus, which we talk about, and it starts from mitochondria. And extrinsic is by the external stimulus. It starts with the tumor necrosis factor, which the other terms is death receptor, and then activators of caspases. When we talk about intrinsic stimulus, mitochondria secrete cytochrome C, and then cytochrome C attached to apoptosis. Now, how this intrinsic pathway carried out, will deal with detail. In extrinsic, there is a tumor necrosis factor is one example of the external stimulant. Now, when a TNF comes and bind to the death receptor, it activates the caspase. Now, again, the caspase which is present in the inside the cell in a proactive form converts to activated form when tumor necrosis factor attached at the death receptor outside the cell. So when there is an attachment of the signal outside the cell, there is cell signaling which will activate the caspase. Now this is the intrinsic pathway. You can see that there is caspase 9, which is an initiator caspase which will initiate the process. Now to this caspase 9 there is attachment of Apoptosis. Apoptosis is a type of, we have studied in the previous slides that Apoptosis is CED4. Then to it there is binding of cytochrome C which was released by mitochondria. So this procaspase 3 which is present in an inactivate form converts to an active caspase 3. Now this caspase 3 is an effector. This caspase 3 will again activate rest of the effector caspases and result in cleavage of the nuclear lamins, cytoskeleton and DNase inhibitor, resulting in apoptosis. Now the same in much detail. We have seen that at the topmost position there is mitochondria which is releasing cytochrome C. Now this cytochrome C come and attach to caspase 9 which has APAF1, and this whole complex is known as apoptosome. Now in the same we can see that mitochondria can also synthesize omine or HR2, SMAC or Diablo. Now Diablo when secreted they are inhibitor of apoptosis IAPS. Now if this type of proteins are synthesized, then they will inhibit the synthesis of cytochrome C. Now once IAPs are, that is the inhibitor of apoptosis, is synthesized, then it will inhibit the apoptosome formation. Once apoptosome formation is inhibited, there will be no apoptosis. Mitochondria will secrete cytochrome C. There is formation of apoptosome which result in activation of procaspase 3 and resulting in cell death. But if there is activation of IAPS, then there is inhibition of the activity of apoptosome. Now the regulators of apoptosis, can see that BCL2 which is CED9 inhibitors of inhibitor of apoptosis. Now these inhibitors are present in mammals and Drosophila but are absent in C. elegans. Now pro-apoptotic means they are not apoptotic. So once they are activated, they will become apoptotic. The examples are BAT, which are multi-domain, and BID, Noxapuma and BIM, they has BH3 domain. These two categories are pro-apoptotic. They normally does not cause apoptosis, but when they are activated, they will cause apoptosis, whereas anti-apoptotic is BCL2 and BCLXL. Now when we see in this diagram, there is a normal cell which has BH3 which is present in an inactive form, and the anti-apoptotic is binding to the multi-domain of pro-apoptotic. Anti and pro, when they have binded, there is no cell death. Now in apoptotic cell, when there is cell death signal, then the BH3 domain has become activated, which was initially pro-apoptotic. Now it will become apoptotic. Now once it has become apoptotic, it will bind to the anti-apoptotic domain and will free the multi-domain. Now once multi-domain has been free, then it will result in apoptosis. Again, whenever there is apoptotic signaling, then the BH3 pro-apoptotic will activate to form a BH3 activate or apoptotic domain. Then that apoptotic domain will bind to anti-apoptotic protein, releasing the multi-domain, and that multi-domain will help in apoptosis. Extrinsic pathway, TNF, that is tumor necrosis factor, which is binded at the topmost to the receptor, activates the caspase 8, which as activations give BID is activated or BAX is activated. These were pro-apoptotic, now they are converting to apoptotic. Once their apoptotic activity has been activated, then they will activate mitochondria to release cytochrome C, which will activate caspase 9 to form apoptosome, releasing the effector mechanism and cell death. This is the extrinsic pathway because the initial signaling starts from the binding of tumor necrosis factor to the tumor necrosis factor receptor. Thank you.